Welcome to this new video about proteins, and particularly today we will talk about enzymes. It corresponds to the chapter 6 of your book, but it comes after the cellular metabolism part that we covered in a previous video. So what are enzymes? We call them biological catalysts because they perform biological reactions or chemical reactions happening in cells. Enzymes work well because of their three-dimensional shape, and they have to have a specific shape to be able to interact with their substrates and perform their functions. So what do we mean by a biological catalyst? The definition or the properties of enzymes are that even if they perform chemical reactions in cells, first of all, they are not consumed themselves by the reaction. So enzymes will do the work but not be themselves affected by the reaction. They basically speed up a reaction that would naturally occur, but probably you wouldn't be able to see the reaction occurring because of the time frame, and the enzyme is here to speed up a reaction that would naturally occur but over a very long time. Importantly enough, enzymes don't modify the equilibrium, so you have the same amount of products and substrates at the end of a reaction that is performed by an enzyme as you would have if the reaction would occur naturally. So the presence of the enzyme will not modify how much you will have in terms of substrates and products at the end of the reaction, but it will simply achieve the equilibrium faster. Um, Also very important is the fact that enzymes will only perform reactions that are already spontaneous. So reactions that are favorable energetically, because enzymes will not modify the delta G. So refer to the previous video if you don't remember what this means. And they will not make a reaction that was endergonic or non-spontaneous occur. In contrast, enzymes will only be able to speed up reactions that would normally and spontaneously occur. So what is their job then? In fact, they work by reducing the activation energy of a spontaneous reaction. What this means is that even if the reaction is spontaneous or exergonic, often you need a first input of energy or a first activation energy in order to be able to counteract certain physical constraints. So, for example, if we look at the next slide, the example that is taken here is a substrate that would be a stick of metal, and the product would be a broken stick. But before you reach the state of the broken stick or the product state, you have to go through an intermediate that is called a transition state, where the stick would be represented as a bent stick. Okay, and this is a state that is extremely difficult to reach. So there is a certain amount of energy that has to be spent in order to put the substrate in its transition state. So if you look at the picture at the bottom, you see that in the enzyme, in the site where the blue enzyme here would take the substrate, it makes a complex called ES for enzyme substrate complex. And what will happen is that if you look at the shape of the enzyme, it's not exactly complementary to the substrate, but complementary enough to be able to take it. Then the substrate will be attracted to fit into the active site by the means of charges and other attractions. And this state is called the transition state. And after that, the enzyme will be able to perform the work and cut the bonds, for example, that keep the stick as a single unit, and therefore produce a product that is made of two pieces of broken stick. So if you consider the substrate, it would be the exact same thing. A normal substrate would have a first state where it is connected with the enzyme, then a second state where it is in transition, and then the final state where it would be converted into a product. So you have to understand that even if the reaction between S and P is spontaneous, it releases energy, or at least it doesn't consume energy, it is required that there is a change in the shape of the substrate before you reach the state of the product. And this is what constitutes a barrier, and it is exactly what enzymes do. They reduce this barrier, they make it possible for the substrate to reach the transition state. So if you represent that in a more scientific fashion, you see the variation of energy in the reaction between the reactants or the substrates and the product. So the reactants at your left, and the products at your right. What is represented in the vertical axis is the free energy. So you see with the blue line that if you don't have an enzyme, or if the reaction is uncatalyzed, so without an enzyme, what happens is that you need to have a very big amount of energy before you reach the transition state. And once you reach the transition state, then it is possible to reach the state of the product. And now the reaction is very easy to perform. But with the enzyme, which is represented with the red line, what happens is that the energy required to reach the transition state is much smaller, so it is much easier, and therefore it is much quicker to reach the state of the product. So how does it do so? It is important to understand that it is the physical shape, the three-dimensional shape, as well as the complementarity of charges and various attractions and repulsive forces that will make it possible for the substrate to interact with the enzyme. So if you look at the left picture, an enzyme is represented, and you see that in the middle there is a kind of pocket produced by the folding of the protein, and the pocket will be able to host the substrate. So the substrate that will come here will fit physically first. Then you will see that there are charges and other possible bonds formed, like hydrogen bonds or van der Waals attractions, that will be possible between the substrate and the amino acids covering the active site of the enzyme. And this complementarity will make it possible for the substrate to fit and to reach the transition state in order to fit better. So the transition state will be represented by a kind of bent molecule, and it will make it easier for the enzyme to process to the reaction, to proceed to the reaction. Now, it is possible that some molecules will inhibit the reaction. It is the case when a molecule is able to mimic the substrate, but without being able to be processed like the substrate. So it is called a competitive inhibitor here. And what happens is that the competitive inhibitor will be able to take the spot where the substrate should normally bind. And of course, if you have the same amount of competitive inhibitors and of substrates, it will be an half and half, so it will be a competition between the two. If you have more substrates, then of course, like what is represented in B, there is more chance that the substrate is taken instead of the inhibitor. If you have more inhibitor, then the chance is more in favor of the inhibitor. So basically, the extent of inhibition will completely depend on the concentration of the inhibitor. You also know that the binding of the substrate and the binding of the inhibitors are always reversible. So even if the inhibitor takes the spot of the substrate, it is still possible that the inhibitor is removed and then that the substrate takes the place. We also often see molecules that can block the enzyme, not by interacting with the active site, so not necessarily by taking the spot of the substrate, but by blocking a change of shape, or by changing the shape of the enzyme so that it's not anymore capable of binding the substrate, or so that it's not able anymore to process or to proceed to the reaction, like to cut or to process the substrate once the non-competitive inhibitor is present. So all the inhibitors that don't bind with the active site are called non-competitive inhibitors. Finally, a last type of interaction is called allosteric molecules, or molecules that bind allosteric sites. And this can give rise to two different effects. You have allosteric activators, and you also have allosteric inhibitors. And these are molecules that will bind with the enzyme, but either make a change of shape that will activate the enzyme, or make a change of shape that will inhibit the enzyme. So here is given an example with threonine, which is the substrate of an enzyme, and the presence, in fact, the enzyme is normally inactive, but when the substrate is present, the enzyme will change its shape and it will become active. So it is an allosteric activator. Then once the threonine is processed, it will end up in a first product that will be converted by a different enzyme, and you will have a cascade of 4 enzymes that will give rise to product number 4, and this almost final product will be converted into a final product that is called isoleucine. And this final product, when present, will be able to combine with the first enzyme, the threonine dehydratase, and it will be able to change its shape in order to deactivate the enzyme. So it is a very efficient means to stop the cascade from start. So this is a typical example of a metabolic pathway where you have one first substrate, several enzymes working one after the other in order to make a final product, and the final product deactivates the first enzyme.